Um, are there any questions?
Hi. For the cancer field, would be very interesting to know, could this be coupled with mutation identification? That's your standard single-cell attack-seq, but acquiring one or few genes, or hotspot mutation.
Yeah, we're developing a platform to try and do that where we, and not just for cancer, I mean, I think that's a great application, but also for things like CRISPR screens, where we can have a whole panel of CRISPR guide RNAs to target different genes, and then actually, instead of trying to sequence the transcript of or the guide RNA, we can actually sequence the location that it's supposed to edit, so it's even a more direct readout than a lot of the perturb-seq type approaches or crop-seq things. So we get, yeah, a much more direct readout, so we could use that for specific mutations in cancer. But yeah, we're excited about applying it to massive CRISPR screens, and especially with the cell numbers that we're going to be able to produce and have started to be able to produce, we'll have a lot of power to really resolve some of the effects of variants on these populations. So, yeah. Mm-hmm.
Thank you for the talk. I wonder if you could speak to co-assay, both single-cell RNA-seq and single-cell attack-seq, since you're doing amplification in your well-based strategies.
Mm-hmm. Yeah, it's an area we've been interested in for some time. There is the Sky-CAR approach, where it's combinatorial indexing co-assay. There's certainly some challenges with that. It's extremely difficult for one, and the data quality is not great. It's more of a proof of principle. And that was more from Jay's lab. In my lab, we've been working on some strategies, somewhat similar to a preprint that recently came out from Kunjang's lab, the snare-seq, which is, I think, really cool. There's also another one that I've heard about from another lab. So there should be a couple coming out in the near term. All of the methods, though, that for attack and RNA that are at least high throughput still suffer from the amount of information generated per cell. So I think there's still a lot of room for improvement. I've heard of some quality data where it's actually just single cell, single well that's been generating higher quality data, but then you suffer the low throughput. So it's a trade-off. I think we've got a little ways to go, but we've got some strategies to move forward towards that, so we can hope.
Thank you.
Hi, great talk. I have two questions. The first one is regarding your Psi attack-seq. Could you briefly mention what's the cost per cell compared to the plate-based or droplet-based sequencing?
Yeah, it's hard. I mean, it depends on the number of cells that you profile. I'll say for a typical prep for, let's say we do a prep that's 20,000 cells, it'll probably cost, and not including sequencing, probably about 500. That's a high estimate, probably US dollars. For yeah, like 20,000, but it's also about 500 for 50,000 or 60,000 cells. So I mean, the cost per cells, I mean, it's more the cost per prep. A prep's going to cost anywhere from $500 to $2,000, but that can, and actually using the CellC approach, it dramatically reduces the cost because you're doing the PCR just on the chip. So for that, it's the cost of the CellC chip and some PCR reagents. It's very cheap because we make all of our own transposase. So yeah, it's hard to say, but I mean, fractions of a cent per cell. It's just it's the sequencing that's the biggest cost. I mean, at the scale we're talking about now, I mean, the cost of doing the prep ends up being negligible because you'll spend, $1,000 on a prep, and it'll be, $100,000 of sequencing. So it's really kind of eliminating the cost of the prep.
My second question is for single-cell methylation data. What's the horizontal coverage that you get with the single-cell methylation in terms of number of CpGs covered by the sequencing?
Yeah, it varies. I think in the published data, it was typically around, our libraries would produce like 2 to 5% of the CGs in the genome, so substantially less than like the single-cell, single-well strategies. But if we keep sequencing more and more, we'd get more. It's just when we have so many cells, the cost of sequencing is really high, and so we haven't gone as deep. We have a newer version of the protocol that we're working on that most importantly makes it a lot easier, because the prep that we published it was very difficult, and it's like a 16-hour prep with no stopping point. whereas this new one, there's multiple stopping points, and and we were able to obtain higher coverage. But I think the best we're going to get with the combinatorial indexing type strategy is going to be maybe 20% of the CGs in the genome if you do extensive sequencing.
But do you still observe this huge amount of chimeric rates in your library?
No, actually, one thing that's nice about the combinatorial indexing approach is the adapter 1 is incorporated using the transposase, and so you don't have the 2 rounds of randomer priming. It's often when you have those 2 rounds that you get the chimeras, whereas when you have a transposase anchor, it dramatically reduces the chimeras. We haven't really detected that chimeras, so it hasn't been as much of an issue. Yeah, I think, but just because we have that transposase-based incorporation of adapter 1.
Thank you.
Hi, I wondered if you have any insight or what your reasoning was to go to the Takara, the isolate platform, rather than adding additional subsequent rounds of classical combinatorial indexing. Is it just a ease of use, convenience thing, or were there other?
Yeah, a couple reasons. I mean, the ways that you can scale things up are increasing the number of wells or increasing the rounds of indexing. So if you increase the rounds of indexing, you dramatically decrease the amount of information content per cell, because the way that you have to add in additional rounds involves chemistry and molecular biology that is far less efficient. And we thought about going that route, but immediately kind of cut it out because we realized even with 2-level indexing, I mean, PCR is extremely efficient and transposase is extremely efficient. So if we can keep it to just those and then just increase the scale with these other platforms, that'll increase our throughput to a number that is, I mean, I can't imagine many instances anyway where we'd want to go beyond multi-million cell data sets. For now, I mean, ask me in like a couple years and I might say that, a couple million is like nothing, right? I mean, the speed that this field's progressing, it could very well be the case, but so we just wanted to have the high quality data. Even if we're sequencing more shallow, we have less duplicates when we have a more complex library. And we decided on the isolate system. So we're using the CELSI system is what I was talking some about. We have some data from the isolate system where we do transposition in their 5,000 well chip, pool everything, and then distribute around 500 per microwell, or one of their microwells. One advantage of their platform is you can successively add reagents to the microwells, which you can't with other microwell platforms. So that's why we were excited about their approach, and that's the data set that we actually have like from last week, where it should end up being probably QC passing just shy of 2 million cells, and it looks like they're also very high quality, like around 50 or so thousand unique reads per cell. So pretty high throughput, and so we'll probably mix between Takara and the CELSI system.
Hi, thanks, a great presentation. I just have a small question. So you mentioned an amazing data set of 2 million cells. Do you have any idea on when it will be available for other researchers?
Hopefully soon. I mean, it's on GM12878, so it's not like it's all that exciting. But we'll probably apply it pretty soon to some of our brain samples. One of the things that we're going to start doing in my lab is as soon as we develop a technology, often what takes a long time is then to get it published, right? You have to apply it to some biological system, and the analysis of that takes a really long time, even though we've had the technology for a long while. So I think we're going to start putting out preprints just as soon as we have a technology that works, and publish the protocols and release all the data in advance. And then we can apply it to some biology and go through the peer review process. But so our goal is to get all of these out as soon as possible. I think the single-cell attack-seq protocol for the CELSI platform, we're hoping to have that released and out to the public in the next month or so. And I would imagine for the Takara one as well. I mean, we just started that project like 3 weeks ago with Takara, and actually 2 weeks ago, and we already have this data set, so it's moved pretty quickly. So we'll probably, but it worked well, so we'll probably try and get that out pretty soon, just because I think people would be interested in that. So yeah, but as far as the data set, I don't think we're going to sequence that sample very deeply, just because it's GM12878, which is not interesting. I'm not sure when we're going to do the next preps.
Thanks.
Mm-hmm.
I have a question.
Yeah.
So at the beginning of your talk, you were mentioning, it's about data analysis. So at the beginning of your talk, you were mentioning that normally attack seek is more like, the attack signal is more like a yes or no signal. But later on, you were talking about calculating differential accessibility and the pseudo-time analysis. So that's more on a more quantitative signal. So which kind of data transformation of analysis strategies you use to
differential
into
accessibility, usually the sites that come up are not accessible in one cluster and only accessible in one or things like that, so it's pretty extreme. And we don't use just the same algorithms as for single-cell RNA-seq for differential expression. It's slightly different. I don't remember the specifics. It's evolved over the last several years. But yeah, that's for that. And then for the ordering and stuff, a lot of what we do is kind of on the global scale. We actually really like to do the pseudotemporal ordering with the topics, which is an aggregation of a number of regulatory loci that we get out of the systopic tool. Because those topics almost always, at least a lot of the top topics, correspond very well with real pathways in the system that we're looking at. So we can actually track it at that scale, and that's often what we'll use in pseudotemporal ordering, or individual sites, but often even with individual sites, through pseudotime it'll be off and then on, whereas the proportion of cells where it's observed just dramatically increases as you go through pseudotime. So it's not necessarily using the counts in each individual cell, but more the number of cells that have counts.
Thank you.
Okay.
Any more questions? No? Okay, so well, we can thank Andrew again.